Hello, everyone. This is Dr. Kuo, and today we'll be starting the first part for our series of lectures on nervous system. So the nervous system is the master controlling and communication system of our body, and every thought, action, and emotion reflect its activity. The cells of our nervous system communicate by electrical as well as chemical signals, which are rapid and specific, and usually causes an almost immediate response. So we begin this topic with a brief overview of the functions and organization of the nervous system. Then we focus on how neurons generate and propagate electrical signals. And then in one of the later lectures, we'll then discuss how neurons work together. Okay. So the nervous system has three overlapping functions. We've got the sensory input. So the sensory component takes in information gathered from internal as well as external changes. And then we have the integration component, which allows for processing and interpretation of sensory input. Finally, we have our motor output component, which allows for activation of effector organs, namely muscles and glands, and that helps elicit, bring about a response. Okay. And so an example would be someone tapping on your shoulder, okay, and you sense it. Someone tapping on your shoulder, that's a sensory input. And then integration, many of the integration actually happens in our brain. So some thoughts run through your head, right? And then motor output, an example of that will be you turning around to see who tapped you on the shoulder. So the action of you turning your head, okay? By the way, these slides may show section figure number different from those in your textbook, so just ignore these numbers. Do use your textbook to supplement your understanding of the concepts discussed during these lecture recordings. Our nervous system is divided into two principal parts: the central nervous system, CNS, and the peripheral nervous system, PNS. The CNS consists of the brain and spinal cord, and it's the main integration and control center of the nervous system. It interprets sensory input and dictates motor output based on our past experiences, based on reflexes, and based on current conditions. On the other hand, the peripheral nervous system is part of the nervous system that lies outside the CNS. So the PNS consists mainly of nerves as well as ganglia. So recall from anatomy, nerves are bundles of neuronal axons, whereas ganglia, okay, are clusters or collections of neuronal cell bodies. So our peripheral nervous system consists of mainly nerves and ganglia. Spinal nerves carry impulses to and from the spinal cord, whereas cranial nerves carry impulses to and from the brain. And these peripheral nerves are important as they serve as communication lines that link all other parts of the body to the central nervous system. Here we have a diagram that shows the organization of our nervous system. As we just discussed, the nervous system can be divided into the CNS and the PNS. The peripheral nervous system has two functional subdivisions. Okay, we've got the sensory, also known as the afferent division, and then we also have the motor, also known as efferent division. Okay, so afferent meaning carrying toward, we're talking about carrying the signal toward the CNS, whereas efferent meaning carrying away, we're talking about carrying signal away from the CNS. Okay, so The sensory or afferent division consists of axons or nerve fibers that convey impulses to the CNS from sensory receptors located throughout the body. Whereas somatic sensory fibers convey impulses from skin, skeletal muscles, and joints. Visceral sensory nerve fibers are going to transmit impulses from the visceral organs. Okay, so such as your stomach, intestine, gallbladder, other organs within the ventral body cavity. The sensory division as a whole keeps the CNS constantly informed of events going on both inside and outside the body. Okay, now the motor division or efferent division of the peripheral nervous system transmit impulses from CNS to effector organs like muscles and glands. And these impulses activate muscles to contract and also activate the glands to secrete. So in other words, they affect or bring about a motor response. The motor division has two main parts: the somatic motor system and the autonomic nervous system. The somatic nervous system is composed of somatic motor nerve fibers that conduct impulses from CNS to skeletal muscles. And it's often referred to as the voluntary nervous system because it allows us to consciously control our skeletal muscles. On the other hand, the autonomic nervous system is also known as the visceral motor system. Okay. It consists of visceral motor nerve fibers that regulate the activity of smooth muscles, cardiac muscle, and glands. Okay. Autonomic means a law unto itself, because we generally cannot control such activities as pumping of our heart or the movement of food throughout our bowels. The autonomic nervous system is also called the involuntary nervous system. And as we will describe in a later class, the ANS has two functional subdivisions: the sympathetic division as well as the parasympathetic division. And typically, these divisions work in opposition of one another. Whatever one stimulates, the other inhibits. So here's a diagram showing the image of a typical neuron. Keep in mind, neurons come in different shapes and sizes. And so this is just sort of a generic typical neuron. And so it's useful to think of neuron as shaped like a tree and containing three major parts. We have the branches called the dendrites. We've got the trunk called the cell soma or the cell body. And then we also have the roots called the axon. Okay, so dendrites are the receptor regions that receive input signal from nearby cells, nearby neurons. And then the cell body integrates that signal. The cell body also contains the nucleus, the mitochondria, and also other cellular organelles necessary for synthesis of proteins. Okay. Proteins including the neurotransmitters that are secreted from the axon terminal. So all that is made within the cell body. And then we also have the axon. This axon is used for transmission of a type of signal, electrical signal called the action potential. So action potentials travel down the length of the axon until it reaches the axon terminal. Okay, and then we'll learn the mechanism through which the action potential then triggers release of neurotransmitters from these axon terminals. Okay, and recall from anatomy, we also have these segments of myelin sheath. Okay, and between the myelin sheath, we have the nodes of Rambier. Okay. So take a moment to review your neuron anatomy. You can watch this video. But keep in mind that information flows through a neuron in a certain direction, and that is from the dendrites across the cell body down the length of the axon and to the axon terminal. So there is a unidirectionality to the flow of signal across this neuron. Okay, what that also means is that you won't see the axon terminal being the receptive region and then transmitting electrical signal in the other direction. So you will not see that. You will always see it only in this direction. Dendrites being the receptive portion, and then that signal then going in this direction. Okay. And eventually, when the signal gets the axon terminal, that electrical signal gets converted into a chemical one, and that's the topic of a later discussion on synaptic transmission. Okay. Many of our neurons have axons that are covered by myelin. Okay. And that myelin sheath, or the development of the myelin sheath, is necessary for controlling our sphincter muscles. And that myelination process only begins, or it begins before birth, but is not completed in the frontal cortex until late in our adolescence. And so oftentimes in newborns and infants, toddlers, right, you often see that they have harder time controlling their sphincters. That leads them to having more accidents in terms of controlling their bowel movement, controlling their ability to hold their bladder. Okay, multiple sclerosis is an autoimmune disease that destroys that myelin sheath in neurons. And so patients with multiple sclerosis have a loss of myelin sheath from their neurons, leading them to have a range of symptoms depending on the type of neurons that are affected. If it affects motor neurons, it can cause speech disturbances. It can cause other problems related to their control of muscles, such as weakness, clumsiness. They may fall, they may not be able to open jars, and they may not be able to control their bowel movement or their bladder very well. But if it affects sensory neurons, sometimes they can also cause sensory symptoms. Okay. Here's the list of terms that I want you to commit to memory. A bundle of neuronal axons in the central nervous system is called a tract. A bundle of neuronal axons in the peripheral nervous system is called a nerve. So that includes most of the nerves you may have studied in anatomy, like radial nerve, ulnar nerve, median nerve, etc. Now, when we find a cluster of neuronal cell bodies in the brain and spinal cord, they're called a nucleus. Outside the brain and spinal cord, we call it a ganglion. So the nucleus is not to be confused with nucleus of a cell. Okay, that's a cellular organelle. But here we're talking about a nucleus, meaning a cluster of neuronal cell bodies. Now, the distinction between gray matter and white matter lies in whether it contains myelin. Okay, so white matter is going to be filled with a lot of myelinated axons, whereas gray matter consists mostly of cell bodies as well as non-myelinated axons. Okay, and so keep in mind that gray matter and white matter is only a distinction that we have as it relates to the central nervous system. Outside the central nervous system, meaning outside the brain and spinal cord, there is no such distinction. Okay, it's all just one color. Okay, and a bit more review on neuron anatomy. Okay, we have sensory neuron, which release information from the periphery to the CNS. here we see it's the spinal cord, okay? It relays information to the spinal cord. And then we also have a neuron that extends from the spinal cord out to the periphery. That's the motor or efferent neuron. And then we have a neuron situated in between the two that helps integrate and relay information from sensory to motor neuron. That's our interneuron. Okay. And then the gap in between them, the junction in between these neurons, those are the synapses. Okay. Let's move on to talking about resting membrane potential. So now, on the topic of membrane potential, there's an electrical disequilibrium that exists between the extracellular fluid, ECF, and the intracellular fluid, ICF. And this electrical disequilibrium is called the membrane potential difference, abbreviated VM. Okay. Sometimes it's called membrane potential. The membrane potential results from an uneven distribution of electrical charge between the ICF and ECF. Okay. So many of our body's solutes, including organic compounds such as lactate, pyruvate, etc., are ions. So that means they carry a net electrical charge. And recall that potassium exists in higher concentration inside the cell, whereas sodium is the predominant cation outside of the cell, okay, in that extracellular fluid. For the anions, chloride mostly remains with sodium, so it's in higher concentration outside of the cell. Okay. Whereas phosphate and many large negatively charged proteins are the major anions inside the cell. So overall, our body is electrically neutral. That means for every cation there is in a body, there's also a matching anion. However, ions are not distributed evenly between the ECF and ICF. The intracellular compartment contains some anions that do not have a matching cation, giving the cells a net negative charge. Okay. And actually, most of these, or many of these, comes from the fact that there are large negatively charged proteins that cannot leave the cell. Okay. And at the same time, the extracellular compartment has missing cations, giving the extracellular fluid a net positive charge. So one consequence of this uneven distribution of ions is that the ICF and ECF are not in electrical equilibrium. Instead, the two compartments exist in a state of electrical disequilibrium. So this slide here just again shows that there's a higher concentration of potassium inside the cell and higher concentration of sodium outside the cell. So if we are to insert ion channels into a cell membrane here, we should be able to see that potassium will flow down its concentration gradient and exit the cell, whereas sodium would enter the cell. In other words, you'll see a potassium efflux and a sodium influx. So this just goes back to an earlier lecture on the concept of passive transport. Okay, and that potential energy being stored in the form of a concentration gradient. So what sets up this membrane potential? Well, this is an idea we talked about in a previous lecture, but here we're going to review it again because this can be a pretty challenging concept for students learning it for the first time. So to show how a membrane potential difference can arise from ion concentration gradient and a selectively permeable membrane, we're going to be using an artificial cell system where we can actually control the cell's permeability to ions and the composition of the ECF and ICF. So in the image here, you can see we've got an artificial cell that exists in chemical disequilibrium, but electrical equilibrium, okay? Meaning that chemically speaking, the ions exist in different ratios across the membrane, okay? We've got potassium that exists only inside the cell and no potassium outside, and sodium only outside the cell, and so on and so forth, okay? But overall, there is electrical equilibrium in the sense that there are equal numbers of charges inside and outside the cell. So there is no membrane potential. Now we're going to insert a lead channel for potassium into the membrane. Okay, and because potassium only exists inside the cell, potassium is going to try to achieve its chemical equilibrium by traveling across the membrane out of the cell. Okay, and as it does that, it's reaching closer to its chemical equilibrium. However, because it carries a positive charge, okay, that also removes one positive charge from inside the cell as the potassium enters into that surrounding fluid. Okay, so that means potassium leaving the cell is going to start electrical disequilibrium. Okay, so now the cell has a membrane potential difference. There's going to be increasing positive charge outside the cell and increasing negative charge inside the cell. So just how much potassium will leave the cell? Well, if potassium was uncharged like glucose, it would essentially diffuse out of the cell until the concentration outside equals the concentration inside. But however, since potassium is an ion, so we have to consider its electrical gradient. Okay, so remember the rule for movement along electrical gradients. Opposite charges attract, okay, and like charges repel, okay? And so as potassium, a positively charged ion leaves the cell, that cell becomes more and more negatively charged. And that negative charge is going to start to prevent potassium from easily leaving the cell. So in other words, the chemical gradient of potassium wants to push potassium out, while the electrical gradient of potassium wants to draw it back in. So for any concentration gradient across a cell membrane, there is a membrane potential difference or electrical gradient that exactly opposes the ion's movement down its concentration gradient. Okay, and at this membrane potential, the cell is at electrical chemical equilibrium, and there's no net movement of ion across the cell membrane. Okay. And so for any given concentration of a single ion, that membrane potential that exactly opposes the concentration gradient is what we call an equilibrium potential. Now, in a real cell, there is cell membrane permeability for potassium that's much greater than the cell membrane permeability for sodium. Okay, meaning we have a significantly larger efflux of potassium through these abundant leakage channels, especially in comparison to the number of leak channels for sodium. So that means there is a much smaller amount of sodium influx. Okay. And so because potassium is charged, as it leaves the cell, it creates a charge difference between inside and outside the cell. Okay. So overall, there's going to be establishment of a negative charge within the cell as the potassium leaves the cell. And at negative 90 millivolts, this is where the two gradients, the chemical and electrical gradients, are equal and opposite. Okay, these two forces are now balanced, and there is no net potassium movement across the cell membrane. So that's the equilibrium potential for potassium. And you'll see that the resting membrane potential of the typical cell is closest to the equilibrium potential for potassium for that reason, because we have a lot more potassium leak channels compared to leak channels for other ions. So this slide just reminds you that there are also a few potassium leakage channels, but they are a lot less abundant than potassium leak channels. So we can consider what happens when we have these sodium leak channels. Okay, which direction is sodium going to travel? Sodium should enter into the cell because it exists in higher concentration outside of the cell, right? So as sodium enter into the cell, it brings positive charge with it into the cell. Okay, and so what that's going to do is it's going to reduce the charge difference between inside and outside the cell from that -90 to -70. Means the resting membrane potential really is a balance of mostly potassium ion leaking out the cell and some sodium ion moving into the cell. Okay, so remember that the number of potassium channels are a lot higher in number than the number of sodium channels. So we can measure the membrane potential that results from excessive cations in the ECF and excessive anions in the ICF. To measure this difference, we can place electrodes inside and outside the cell. And so this shows that in the absolute charge scale, the ECF will be +1, ICF will be -1. In real life, since we cannot measure absolute number of ions, so we have to set the extracellular fluid as the ground as a default 0. So that gives the ICF a relative charge of -2. So we know that many of our toys and electronic gadgets need energy in the form of batteries. Okay, so by separating negative and positive charges, a battery can store potential energy, and essentially potential energy, if you recall, results from the location of the position of an object. Neurons also separate positive and negative charges across their cell membrane. So that's separation of charge is what creates the membrane potential. So there is an accumulation of positive charges on the outside surface of the cell membrane and accumulation of negative charges on the inside surface. And the difference in charge is called the resting membrane potential of the cell. So like batteries, our neurons store potential energy, and neurons use that potential energy to communicate with one another by generating nerve impulses called action potential. Okay, let's recap what we just learned. So higher membrane permeability to potassium than to sodium means more potassium efflux than sodium influx. So that means more positive charge leaving cell than entering it. And that means net negative charge inside the cell. Now, we just discussed how the membrane potential is established. Okay, so how it is established. Now, here comes the next part of the question. Once that membrane potential is established, how do we maintain it? Okay, so consider the fact, or consider the situation where we don't have a mechanism to maintain that higher concentration of potassium inside the cell and higher sodium concentration outside the cell. What happened is that the leakage of the two ions would eventually cause the concentration difference between outside the cell and inside the cell to disappear. Okay, so this is where the sodium potassium pump enters the picture. So this enzyme called sodium potassium ATPase, or sodium potassium ATP pump, maintains the concentration difference of sodium and potassium across the membrane. Okay. So in a cell, energy come in the form of ATP is used to drive this pump. So for each cycle of the pump, it results in 3 sodium ions moving out of the cell and 2 potassium ions moving into the cell. Okay, and so the activity of the pump causes extracellular sodium to be higher outside the cell versus the cytosol, and intracellular potassium to be higher than outside the cell. Okay, and so to recap this, sodium potassium ATPase is an enzyme and a carrier protein that moves 3 sodium ions out and 2 potassium ions in for every ATP molecule used. And this next slide just recaps the 2 concepts we just learned, which is that the resting membrane potential being negative is established by higher membrane permeability to potassium than to sodium. Okay, and then the maintenance of that resting membrane potential is carried out through the action of the sodium potassium pump because it helps maintain higher concentration of potassium inside and higher concentration of sodium outside the cell. And these next 2 slides have a link. Please use these to review the concepts that we've just learned. So these are animations discussing the resting membrane potential. So take a moment to watch these to familiarize and make sure you understand these concepts.